webinar hosted by IXICO. I'm just going to cover a few admin points and then I'm going to hand you over to your moderator and chair, Robin. So just to let you know that there is a question section. If you look to your right, top right of your screen, you'll see a question mark icon. And under that, you can ask questions anytime through the session. We do have a session at the end dedicated to Q&A. So we will hold questions till then, but we'll moderate, look at your questions and then come back to you. Because we've only got an hour, we want to be respectful of people's time. So we may not get to all of the questions. And if the questions we don't get to, we will respond to you directly. May I also just direct you to, there is a captions icon at the bottom right of your screen, which enables you to just see the text as it's being spoken, if you would like to turn that on too, and if it suits you to do that, if you're in a noisy background, for example. But otherwise, I will hand you now to Robin Wolz, who's Chief Scientific Officer at IXICO, to begin the webinar.
Thank you, James, and welcome everyone to this webinar in our Huntington's disease series. I'm delighted to host you through today's sessions that focuses on two critical topics in HD trial design. How do you select the right endpoints for HD trials, and how can advances in neuroimaging drive better trial outcomes? Over the past years, we've seen significant momentum in the HD trial landscape. We have many promising drug candidates in development now across several therapeutic modalities. We see trials with small molecules, biologics, ASOs, gene therapies, just to name the most prominent ones here. We also see different mechanisms of action under development. And so each trial comes with its unique regulatory pathway, expected effect sizes, and trial design requirements. So a key question in the field is continuing to be around the most meaningful clinical endpoint to measure treatment effect, most generally, and then also specifically for the different trial designs. I've just mentioned different populations and so on. Separately, we see continuous increase in our understanding of different biomarkers, how they tie to the biology, and how they can be deployed in the most efficient and robust manner in clinical trials. As part of today's webinar, I'm very excited to hear from two distinguished experts in the field on the latest insights on both topics and how that knowledge can be used to improve trial design and ultimately decrease the time it will take to get effective drugs to patients. So without further ado, please let me introduce today's speakers. Dr. Jeff Lang is Professor of Psychiatry and Biostatistics at Iowa University. Jeff is one of the leading statistical experts in HD clinical trial design. He works closely with the CHDI Foundation, several companies developing drugs in Huntington's disease, and he's a co-chair of the CPAF's HD Regulatory Science Consortium Modeling Working Group. Jeff's presentation today will focus on how we measure, what we measure, and how we improve on what we measure. Separately, I'd like to introduce Dr. Marina Papoutsi, who is a senior biomarker scientist at IXICO, where she leads the scientific activities in Huntington's disease. Prior to joining IXICO, Marina was a senior research fellow at the UCL Huntington's Disease Center, working with Professor Sarah Taprisi on the development and validation of neuroimaging biomarkers in Huntington's disease. Marina will present current best practice methods in imaging for HD trials, as well as results on those new analytical approaches. And with this, I'd like to hand to our first speaker, Jeff Lang.
Thanks, Robin, for that introduction. I'm excited to tell everybody about some of the work that we're doing at the University of Iowa regarding endpoints in HD clinical trial planning. Next, please. So as Robin mentioned, I'm just going to briefly talk about who we might measure when planning a clinical trial. And I'm going to concentrate mostly on what we intend to measure and how we might improve the measures that we select. Next. So in terms of who do we measure, well, the recommendation is to start with the HD integrated staging system, which you see there with its 4 different stages. And today I'm going to focus on stage 2 and into early stage 3. This is the recommended interval of progression that might be most appropriate for current clinical trials, in that clinical variables are moving, but people have a reasonable amount of quality of life that might be preserved via treatment. After starting with the HDISS, then further enrichment can be performed using one of the prognostic indices in HD, which is either CAP 100 or PIN. Next, please. So once we identify our treatment population, then what do we measure? And today I'm going to focus on efficacy outcome variables. And there's 2 classes of candidates, the biomarkers and the clinical variables. In terms of the biomarkers, we have things such as caudate volume, perhaps whole brain volume, and then fluid biomarkers such as NFL, PANC, etc. Then we have our so-called usual suspect clinical variables, total motor score, symbol digits, Stroop word, total functioning capacity, and then the cUHDRS, which is a combination of the first 4 of those variables. Next, please. So therapy modality is important for HD clinical trial planning, and I'm making the distinction here between small molecules and cell and gene therapies. With small molecules, at least historically, this has involved randomized controls, larger samples, and with gene therapies, this has historically involved external controls with smaller samples. So with that background, we can go to the next slide. And I want to give you a summary of thoughts about both treatment and outcomes that come from a series of workshops that are sponsored by CHDI. These are known as the Path for HDISS2 workshops. And we've had 2 meetings so far in London and Nashville, and there's going to be another one later this year. And the recommendations are the following, and again, distinguishing between therapy modalities. For small molecule biologics, it's thought that the HDISS2 would be the best treatment population stage with enrichment based on CAP 100 or PIN, as I mentioned. Novartis has this, or is consistent with this in their protocol, if you want to take a look at clinicaltrials.gov. For advanced therapies, we might go a little bit further into early stage HDISS3. And so there's other sponsors that are considering this progression interval. Today, when I show you the natural history data, I'm going to be considering the later HDISS2 to early HDISS3. In terms of primary endpoints, the biomarkers from this workshop were not thought to be ready for prime time in terms of primary efficacy endpoints, but they are very important for safety. And Marina in her talk is going to address this in terms of brain volumes. The cUHDRS is most efficient, as we're going to see. But the TFC arguably is the most clinically meaningful, and the sponsors, as you see there on the right, are using the cUHDRS as a primary and TFC as a secondary, or some variation on that theme. Next, please. So I always think it's instructive to look at the natural history of all our candidate variables. And so here we have panels of the different variables. And in each panel, the individual trajectories are denoted by these thin dashed lines. And the thick black lines are model-based linear change over time. On the horizontal axis, we have year of study. And this goes out to about 12 years for enroll, and a little less for the other variables. On the left, we have imaging variables, which come from the IXICO GBSI pipeline, and are from the 3 studies that you see listed there. For the clinical variables, the data comes from Enroll HD, which has thousands upon thousands of participants, as you probably know. What I want to draw your attention to here is that the caudate and whole brain trajectories are relatively regular compared to the natural history trajectories. And this has implications for trial planning purposes. In addition to the regularity, we have, say, a slightly faster rate of change for caudate than whole brain, as you can see there. And we have different rates of change for the clinical variables, as you can see in the right panels. Next, please. So what I did was to just give a rough estimate of a total sample size for a randomized controlled trial. And so in the graphic on the left there, you can see the measures are listed at the bottom, and then I've ordered the variables in terms of the total sample size that we would require for a clinical trial. And I've rounded these numbers. I am considering a two-arm trial, so with equal allocation, and a 3-year duration, a 40% treatment effect, the standard levels for type 1 error rate and power. And I'm not considering a placebo response, which you probably should do when planning a clinical trial. That would make these numbers go up a bit. So next, please. So the thing to notice here is that caudate and whole brain give very small total sample sizes. Again, this is related to the regularity of the natural history, as we saw in the last slide. Next. In contrast, we have the cognitive variables, and so SDMT has a lot of variability in the enrolled database, which leads to the highest total sample size. And next. So our winner, if you will, in terms of the sample size competition would be the cUHDRS, which again is the combination of the variables for the last 4 bars there. And so the cUHDRS does give a smaller sample size, and so it may be the best of the clinical endpoints that we have right now. So when looking at the totality of this figure, the question is, should we use imaging? And sponsors probably would want to use imaging because it would lead to a much smaller trial. But there's been a problem in that the clinical trials that have recently finished have not shown consistent signals in terms of imaging the clinical variables. Some studies have shown positive efficacy signal for clinical variables, but not for imaging, and vice versa. So I don't think that regulators are going to accept caudate volume, for example, as any kind of surrogate or intermediary variable until we find evidence that there's consistency in these signals in actual clinical trials. So I think for the moment, we're wedded to a clinical variable, and probably this cUHDRS is the best candidate because of the efficacy as illustrated in this figure. Next, please. So we want to use the cUHDRS, but there's historically been some pushback about this measure, especially from the FDA. There's 3 objections that I think are rather easy to address. One is that it's misnamed. If you look at the FDA guidance, if you're combining together continuous variables as we are in the cUHDRS, it should be called the combination UHDRS, not the composite UHDRS. Composite being reserved for multiple time to event variables. The formula uses z-scores, so you can see there TMS. You subtract out A, which is a mean, and divide by B, which is a standard deviation. And the FDA historically is not like this because you lose the meaning of the original metric when you use these scores. And finally, this was an industry, the original cUHDRS paper was an industry publication that was sponsored, and the first author was a sponsor employee. Next. So the 3 more challenging points here are the following. The cUHDRS is a combination of different domains, motor, cognitive, and functioning. And if you look at the FDA guidance, it would be preferable for each of the components to be similar in importance, which may not be the case. For example, regulators may favor functioning over motor and cognitive. And they have to have similar movement under treatment, and the it remains to be seen if this criterion is met. Furthermore, there's placebo responses that vary across the components. So the analysis that I'm referring to here is of the GenHD1 placebo arm, which showed that when there is a placebo response, that it tends to have a quick onset and tends to be sustained, at least under treatment. But there's evidence that this varies by component, which is not consistent with number one there and the FDA guidance. Finally, the clinical meaningfulness is not clearly defined with the cUHDRS, and I have that in bold because that's what we're working on in Iowa, and I'll give you a little taste of the things that we're considering in the following slides. Next, please. So there's at least 3 paths forward. We can adopt an alternative existing measure, or we can try to improve the acceptability of the cUHDRS, or we could adopt or develop new measures. Now, in regard to one, on the next slide, probably the best candidate for adopting a measure that already exists is the TFC, because that's a feels function survives variable, which is usually favored by regulators. In the aforementioned placebo effect study that I discussed, there was small to no placebo effect for TFC, which is also has merit, and there's arguably inherent clinical meaningfulness in the measure. Now, the big drawback is that you have lower statistical power than the cUHDRS, and the TFC can't be used for early stage 2 trials, for example, because it's not moving. You have to have movement in order for treatment to deflect natural progression. Next, please. So another tact here is to use the cUHDRS and try to improve its acceptance, and this can be done in various ways. We can improve messaging about the science. There are reasons why HD would affect multiple domains. We might improve the validation. What I'm going to talk about today is improving clinical meaningfulness. And 2 concepts that I'm going to discuss are time saved with treatment and responder analysis. Next. So time saved with treatment comes to us from Alzheimer's disease research, and it translates a mean difference into a time difference. So it attempts to answer this research question: how long is the time delay in progression under treatment? And this, of course, relative to the control. And this time delay concept is an interpretation that seems to align well with stakeholders. So as an example, I'm going to present the model-based means of the AMT 130 trial, which have been publicly presented. And we're going to assume here the cUHDRS is used as our measure of progression. And as you probably know, there was 75% slowing in a 3-year trial. But the question is really, what does this mean? And we want to know what this means in terms of a time delay. And what I'm going to show you in the next slide is that we do have a time delay of up to 2 years under this scenario. So what I have there at the top is just a restatement of the research question: how much earlier would the control group, which here is in red, reach the same level of worsening as the treated group in blue? So next, if you just look at the horizontal axis, we have here the cUHDRS, which are model-based means. And if we go to the next image, both groups start at 13.4 because they were well matched, as you probably know. And at the end of the trial, we had the treatment group having a cUHDRS mean of 12.9, and the control group mean having a mean value of 11.5. If we go to the next image, this is a vertical distance of 1.4, which is probably difficult to interpret. A little better interpretation is we have 75% slowing for baseline, but we still want to turn this into this concept of a time delay. And the way we do that is we start with the last mean value of the treatment group there at 36 months, which is 12.9. And our question here is, when did this value of 12.9 occur for the control group? And so we just trace back with a horizontal line, as I have there, until we find that value of 12.9 on the control curve. And then we drop down to the time on study, which we can see is 12. And so if we go to the next image, we have a simple calculation of just the difference between when this value of 12.9 occurred for the treatment group and for the control group, and the difference is the delay of the treatment for this value, which in this case is 24 months or 2 years. Next. So our interpretation here would be that in a 36-month trial, the decline for the treated group was reached at 12 months for the control, so that there's 24 months gained or saved from progression by treatment, or treatment was delayed. The treatment delayed natural history progression by 24 months. Next. So this same analysis can be performed for the TFC or all the components of the cUHDRS, and there's methods for confidence intervals and testing. And note that this standardizes all the variables to a time metric. So this can be a basis for an alternative to the cUHDRS, but I don't have time to discuss that today. Next, please. So that was time saved with treatment, which is a method that works on the means of the treated and the control group. There's another approach called responder analysis, where we start at the individual level and we score individuals in terms of this concept of MCID. And MCID here in this context means the amount of change on the components of the cUHDRS that bother a patient. Now, what do we mean by bothering a patient? Well, we have to have enough change so that either their quality of life decreases to the point that a patient doesn't like it, or that their functioning declines to that point as well. And so there's been two recent approaches to anchoring of the change of our cUHDRS components, and you can see the papers there. I'm going to offer a quality of life illustration today, and I'm going to consider the total motor score and the MCID over 3 years for people in stage 3 of the HDISS. And the MCID threshold is 9.5 from the paper. And this means that a total motor score change that meets or exceeds 9.5 constitutes meaningful progression over 3 years. And we can illustrate this in the next slide. We start with the data for a person over 3 years, and this is an actual participant from Enroll HD. And we could either use these observed means, or in the next image, we could use a fitted model, which would be the dashed line. It doesn't really matter. If we go to the next image, the point here is that the change for this individual from year 0 to year 3 is 17. And the question is, does this exceed the threshold that we set on the last slide? And we already know the answer is yes, but we can show this graphically. In the next image, I show the red line here being the threshold of MCID, and I'm calibrating that to the start point of this individual at year 0. So the black circles would have to exceed at the end of 3 years the red line, and that does here. And if we go to the next image, just to remind you, this is a 9.5 is our threshold. And then if we go to the next image, there's a couple of things we can do in scoring. We could either scale the change in TMS by the MCID, or we could use a binary scoring, just noting whether a person exceeds the threshold or does not exceed the threshold. And if we go to the next slide, you can see, get a taste for how this might be used in a clinical trial. We have individuals in a treatment group on the top row and individuals in a control group on the bottom row. And each one of these are binary coded as to whether they have meaningful progression, which is a 1 in red, or don't have meaningful progression, which is a 0 in green. And so you can see that we can do things like compute the mean of these binary scores across the different groups. And here we can say things such as 60% of the people in the control group had substantial progression over time, whereas only 40% of the individuals in the treatment group had substantial change over time. So this can be done for all the components and can give us insights into the behavior at the individual level of the different components of the cUHDRS. And it can also, of course, be done for the cUHDRS directly. Next. We won't have time to talk about new measures, so I'll just go to the next slide and give you final thoughts. So, it's a struggle to have adequate statistical power in HD studies. The CUHDRS is our most efficient outcome, but it's problematic for some of the reasons that I mentioned. So perhaps time saved and responder analysis can help, but there's ongoing work here to consider things such as statistical power of competing methods. Two design factors that you should know about that I'm not going to discuss are an enhanced control group and a baseline prognostic score. And the work continues in these entities that I have listed there in the bottom. And if we go to the last slide, I just want to thank you for your attention, and we'll have a question and answer period later. Thanks.
Thank you, Jeff. Great presentation. Really appreciate it. There's a couple of questions that are coming in, but as you say, I suggest we take them all together at the end. I'd encourage people to keep sending them, and we'll, in the meantime, hand to Marina for her presentation. Thank you.
Thank you, Robin, and thank you, Jeff. That was excellent. So we're now going to shift gears and we talk about imaging. So IXICO is an imaging CRO, and we support the majority of HD clinical trials. So I'm just going to focus this talk for the next 10, 15 minutes into some of the recent developments in imaging analytics. Things move pretty quickly in the field of technology, particularly with the advent of AI deep learning methodology. So I thought this would be a good opportunity to talk some of these recent developments. So as Jeff kind of briefly mentioned in his talk, so imaging has a very well-established role in clinical trials, even though we're learning still a lot about how to use it in clinical trials context. The most commonly used modality is volumetric imaging using structural MRI, and that's used for safety, eligibility, and also for efficacy, usually as an exploratory endpoint, but also in some cases a secondary endpoint. For eligibility, Jeff mentioned the HDISS staging, so coded imputation volume are landmarks for stage 1. And then for efficacy, coded imputamen volume, particularly coded volume, is one of the most sensitive biomarkers. Other modalities that are commonly used is a diffusion MRI and QSM, quantitative susceptibility mapping, and these are mainly used as complementary metrics to understand more of the effects of treatment in the brain. So as I mentioned, so volumetric MRI is the most important one, is the endpoint that's most commonly used to look at changes, particularly focusing on the caudate. So the caudate is structured deep in the brain, and it's really measuring caudate volume change, highly reliable and sensitive, and suitable even for stage 1 patients, even though most of the trials, the trials so far don't use stage 1. Thinking about the future, for example, preventative trials, it's a really sensitive metric. The method most commonly used, and the current what we call the best practice for measuring caudate volume, is using manual segmentations of the caudate at the baseline. So you can see here on the left side an outline, a green outline highlighting the boundaries of the caudate, and then using a method called the boundary shift integral that looks at the changes in the intensity within that zone to understand how the caudate volume changes over time. That's the method that was developed at UCL. So Nikki Hobbs and Rachel Skyhill have worked extensively on that, and was using the track on HD papers. So it's really sensitive, and it's a very robust biomarker, but it has some challenges, particularly when it comes to scalability. Using manual segmentation of the caudate can take up to an hour doing manual segmentation, so it's not a very scalable metrics, and it's also quite an old method now, more than 10-15 years old. So it is time for a revamp, and we've done a lot of work to try to develop modern methods using deep learning tools to automate the segmentation part, to training algorithms on the manual segmentations, and also kind of the longitudinal part. And we recently completed a very detailed validation comparing different tools to understand the sensitivity of the method. And on the top here, you can see a voxel-wise map of change across the brain. So blue looks at atrophy, red shows increasing volume. And this is a method called Jacobian, and we've developed a deep learning-based Jacobian method that's really fast and efficient, but also really, really sensitive. And the advantage of this method is not just the scalability, but it also is the fact that it can be used for all regions of the brain. So the BSI, the boundary shift integral, can only be used in regions that have very strong contrast. So regions like the caudate that is next to the lateral ventricles or the whole brain. So anything that's close to CSF. But for regions like the putamen, for example, you won't be able to use the BSI. And that has always been a limitation. The putamen, looking at change the putamen has always been quite challenging just because of the contrast in the methods. But so in the figure here, what you can see, we've done this validation comparing different methods, the manual BSI, the IXICO, the GBSI, which is an automated version of the BSI, and also free surfer longitudinal. This is just done using data provided by CHDI. And as you can see, all the methods provide maybe similar estimates in terms of the mean, but what kind of changes is the variability. As you can see, the Jacobian has really much lower variability, and that increases basically its sensitivity. So when you look at the table here, it just looks at effect sizes for 1 year change from baseline for the caudate nucleus. So the Jacobian outperforms all other methods, including the manual BSI, considerably. And similar for the putamen, when you compare the Jacobian to free surfer, it outperforms. So this is one of the ways, for example, when Jeff looking at improving things for the future, maybe moving to some of these modern methods that have a lot of advantages, both in terms of scalability and sensitivity. Moving on now to one of the issues that has come up a lot in clinical trials in HD, and we're constantly talking to sponsors about, is the increase in lateral ventricular volume. So on the top here, you can see figures from the Generation HD1 papers that kind of show these issues, but this is not just for tominersen. We've also seen it in branaplam, and it's been reported in other clinical trials. So it's one of these issues that sponsors are all very aware of and all try to mitigate or try to understand. So basically what the observation is that you have these dose-dependent increases in lateral ventricular volume change. And these are not accompanied, in this case, particularly for tominersen, by similar changes in the whole brain. If you can see here in the natural history, the lateral ventricle volume and whole brain volume are very, very closely related. They have a correlation of 0.7. And this is the change, not just the volume. So lateral volume, ventricular volume change and whole brain volume change has a very, very high correlation. But this is not really, you can see in tominersen, this relationship is disrupted. So to try and understand what's going on, it's important to keep in mind that lateral ventricles are only one part of the ventricular, the CSF space. So when you look at CSF, you've got the lateral ventricles, but there's also the third ventricles, the fourth ventricles, and also subarachnoid space. These are all areas where CSF flows. So when we look at lateral ventricles, we see them increasing, but we don't know what actually is happening in the rest of the brain. So looking at total CSF volume is another interesting endpoint. So looking at changes within the subregions of the CSF and also total CSF. And one of the hypotheses that is currently kind of being considered is whether there is a redistribution of CSF. So maybe the lateral ventricles increasing, but some other areas of the CSF are decreasing to kind of make up some of these changes. So there's a change in the dynamics of the CSF, but not necessarily in the whole volume of the CSF. And we know the total CSF volume changes in HD similar to what we know about the lateral ventricle. So it increases as the disease progresses and there's more atrophy, total CSF volume increases. And this is a figure from Predict HD. In yellow is a control group, and then low, mid, and high cap groups are in red, green, and blue. So this is another interesting endpoint to consider, to try to capture a lot of these changes that are happening in CSF. Another new endpoint, and something again that's there's a lot of recent developments, is looking perivascular space. So CSF flows obviously around the arteries and veins. It's part of the perivascular space. And even though the total volume is very small, we know in HD, particularly for the basal ganglia, you can see here in the top right, there is a lot of vasculature crossing the tail of the putamen, and this does change with disease. So at the bottom here, this is a recent paper by Yin et al. Looking at percent PAVS volume change in the basal ganglia. You can see in controls and then pre-HD and manifest HD that you have an increase in this volume as the disease progresses. This is a cross-sectional study, so it's not actually looking at change over time, but it's looking at change within each group. And another way of looking at perivascular space is using diffusion. So a new method that's being used a lot in Alzheimer's disease is called DTI along the perivascular space, or DTI ALPS. And here you basically use diffusivity along the veins in the deep white matter to understand how, as a proxy, sometimes it's used as a proxy of lymphatic function. And you can see here again in the same paper, Yin et al., that you can see the DTI ALPS index decreases with at the different HD stages you have for pre-HD and manifest HD. So these are some new methods that haven't been tested in clinical trials, but this is something worth considering, and we can talk about it if you're interested in using them for your trial. So moving on now, I mentioned another of these methods that you use in clinical trials, is quantitative susceptibility mapping. Can I take it up? There we go. Sorry about that. And this is a really, it provides really nice contrast in the striatum. We can see the images here on the left, that you have a really strong contrast in the basal ganglia, particularly in the globus pallidus, which is a region that you don't really see very clearly in a 3D-T1 weighted image that's not usually used for volumetric analysis. And even though we know the globus pallidus is affected in HD, we've always been struggled to look at to use it as a biomarker because the contrast in a 3D-T1 weighted image is so poor. But with QSM, we actually have a really nice, strong signal. And you can see here in this paper by Van Bergen et al. That you can actually look at changes over time in the caudate putamen and also in the pallidum. Particularly, I think for the pallidum, if you're interested in areas like the pallidum, the QSM is a really nice method to use. And it is basically the signal from the QSM is a proxy for iron concentration. So as disease progresses, the signal increases. As there's more iron released extracellularly, you've got an increase in the concentration and the QSM signal. And last but not least, we're talking about endpoints. I'm going to talk about PET traces. There's a question that we get very often: is there a PET trace that we should be using in HD? As most of you probably know, there's no mutant Huntington traces available yet, even though there's considerable work being done by CHDI and their collaborators. But there are many options for functional tracers. For example, FDG PET and DSPO have been used in clinical trials, like in Signal HD and in Legato. And there are some promising traces like PD10A. The main stumbling block when thinking about using traces like PD10A is that they are research traces, so they're not commercially available, which makes their availability really challenging in using in clinical trials. And also, if they don't have a validated SUVR kind of analysis, then you're looking at a dynamic scan that can take up to 90 minutes, or doesn't make it suitable for clinical trials. So for clinic, we ideally need static scans for SUVR analysis, F18 tracers that have a long half-life, and then commercial variability to ensure that it can be distributed widely across different sites. In the absence of that, and if you're really interested in research traces, then sub-study, we usually can recommend sub-studies and identify sites that can produce and have experience with these traces. CHDI has funded, and there's a study called Biomarker that's currently running to understand, evaluate the sensitivity of 4 different traces, including PD10A. So which I think will be really useful, because even though we know a little bit about some of these traces and how they work in HD, we haven't really compared them head to head to understand which one might be more sensitive. So I think that would be really a very important first step to identify which one would be the most suitable tracer going forward in clinical trials. And to finish my part, one of the things that we recently completed is the analysis of a very large natural history data as part of the HDIH consortium, the HD Imaging Harmonization Consortium. This is a joint initiative by IXICO Pharma and CHDI, where we aim to reanalyze, track, predict, and image these datasets in a regulatory grade way so that they could be used for filing purposes. So we've basically reanalyzed all the data in exactly the same way, using exactly the same process that we would do for a clinical trial, and using our more updated methods. Like in this case, we use the IXICO generalized BSI method, and this is data that Jeff included some of in his talk. And this can now be used for clinical trial planning, but also to supplement control groups, for example, in hybrid designs. One of the questions we often get, obviously, there's different studies included in this data set. Track and predict were done very, very differently, and the sites very different way of collecting data and harmonizing across sites. But one of the advantages of using some of these modern methods like IXIC-GBSI is that these deep learning methods have been trained to account for some of this variability that is due to differences in image quality, and they're actually quite robust. So here we see here on the right-hand side some of the work that we did using propensity score weighting. So once we adjust for baseline characteristic for different participants from track and predict, actually the slopes are identical. So this is a really important method as well, that it's not just about collecting the images, and the quality of the images is really, really important, but also the way that the images are analyzed and created are really, really important to make sure that they can be comparable, comparability between studies. And on that note, I will finish my talk and pass it back to Robin. Thank you so much for listening.
Thank you, Marina, for this wonderful presentation. Thanks again, Jeff. Great presentations from both of you. And that's also reflected in the fact that we are getting in a lot of questions now, which just shows the excitement by the audience, I guess. So I'll mentally sort them briefly and then maybe start with some of the more general questions before we go into the detail. One for Jeff, you have outlined the FDA's concern around CUHDRS combining different domains. So in your experience, what are the likely consequences of this? So I guess, what are the steps the field needs to take to reassure the regulators or to change direction, so to speak?
I think the most important thing is to demonstrate in an actual clinical trial that the components of the CUHDRS move in a similar direction, which is plausible. And if they were to move the same amount, that would be a bonus. But when we say move the same amount, you need some kind of standardization. And so something like showing the same time saved, as I presented here, would help the situation, I think. Okay,
that's great. So basically showing that actually we're measuring different domains, but essentially they're as meaningful, and therefore the composite makes sense to as an outcome to the trial. Is that the way?
Well, in that the components have a similar behavior under treatment.
Right. Yeah, exactly. And it actually gives me a bit of a segue into the next question, which comes back to imaging. I think you said on your in your presentation, we're not quite there yet to consider imaging as a surrogate endpoint. You pointed to the lacking or the inconsistency in trial findings between what we see on imaging versus clinical outcomes. So it's in a way, I guess, a similar problem in that we need to show that things are pointing in the right direction. So the question that we have here, what data is needed to get a better understanding of the relationship between clinical decline and biomarker change, i.e. move us closer to a surrogate endpoint?
Right. So again, in a clinical trial, you would have to show the direction of signals is similar between clinical variables and imaging variables. And as I mentioned there's been formal presentations summarizing recent clinical trial results, and there's just been inconsistency of the imaging results and the clinical results. Sometimes one is positive for efficacy, and at other times, in other situations, the other one has been positive for efficacy. So until there are probably several clinical trials showing consistency in the direction of the efficacy signal, these variables, until that happens, I don't think that there'd be any kind of intermediary status for imaging or any other biomarker for that matter.
Right, thanks. This seems to be a topic of high interest to the audience. There's a kind of like related question I just saw come in. Do you see value in using cordate MRI as an efficacy biomarker to support go/no decisions after an 18-month phase 2 trial?
The answer in general is yes, but I don't know really what the person means by go, no go. That could be internal to an organization, or that could be more formal evidence presented to a regulator. You certainly want to image if you can afford it, to be frank, for safety, as Marina discussed, but also because if you do have a treatment that shows an effect for imaging and the clinical biomarkers, that would be a very high level of evidence to me of efficacy.
Okay, thanks for that, Jeff. I'll maybe go to some of the more technical questions for Marina, and then we come back to some of the more general questions on trial design. Marina, you've presented some of those novel techniques. You presented on DTI, you presented on QSM. A question by the audience is, how close are we to those being trial-ready endpoints, and what validation steps remain?
So we're already using them in our trials. The evidence compared to, let's say, cordate and volumetrics are much lower from natural history studies, right? But we are already using them in clinical trials and as exploratory endpoints to try and understand how they behave, again, in a clinical trial setting, and their sensitivity, because what we see happening when we move to disease-modifying therapies in the clinic is that a lot of the imaging endpoints don't behave as we expected them to do, and Jeff kind of alluded to that as well. So it's worth including some of these exploratory methods, especially when setting up a phase 1 trial, to see how they respond to the treatments and some of these challenges, but also some of the benefits, and then determine later down the line in the development which ones to move forward or not. But they are clinical trial ready. We just don't know. We just don't have enough evidence to know yet how they will respond to different treatments.
Yeah, that's interesting, and actually use that response to go to what I think a related question, which is, when you say harmonization, what harmonization methods were used? And the reason I think that's related is because maybe you can also comment on what additional burden, I guess, we are putting into the trial by adding some of those exploratory endpoints, because you just well characterized how we can put those exploratory new methods into trials, collect the data, and then essentially build the evidence we need to increase the role of the trial. But that, of course, comes with operational challenges, potentially harmonization. So maybe if you can capture those two aspects in one go, what is the level of harmonization that is required, and what is the extra hurdle, I guess, we need to go to add some of those additional endpoints on an exploratory basis?
I mean, in terms of harmonization, the most important one is done at the scanner level. So when we set up the trial, to make sure that the sequences across the different manufacturers, because different manufacturers have different ways of setting certain things up, that they are as close aligned as possible. And then the second level is the processing, so the data handling, how the data are processed and analyzed, and using a centralized processing that kind of takes away that additional level of complexity. But I think in terms of the burden, obviously an extra sequence is an extra time in the scanner, an extra burden for the patient. So, you know, you can't just be scanning forever. You need to consider what is useful for a specific trial and specific treatment. And we can advise a lot on that based on our experience of what we think will respond more or will be more useful for a certain trial. But yeah, it's additional burden for the patient, particular additional costs in terms of scanning time, and then the complexity of setting up a trial that has more sequences. So the more advanced the sequences, obviously that increases the complexity of qualifying a site, particularly if it's a site that is not very, that's not a research site. So research centers have a lot of experience with a lot of these advanced sequences, but a clinical site might not have so much experience. So that's also something to consider. And again, we can support with all these issues.
Great, thanks, Marina. There's actually two quite specific technical questions I'll briefly pose to you and then come back to another theme, so to speak, for more general questions. So that's a question about PET tracers. What about C11, CHDR, 180R as potential PET tracer for HTT?
Unfortunately, it didn't perform as well as we had expected. There was a paper recently by Aline Delva and the colleagues. But you know, it's not the end story for mutant Huntington. I think there's a lot of work that's been done by CHDI for the second generation tracers, and there were some recent papers published on animal models, not in humans. But I believe it will soon move to humans. So I think this is definitely an area we're monitoring, and we're keen to see how it will evolve, because it will be a game changer, obviously, having a mutant Huntington tracer. But we're just not there yet, unfortunately.
Great, thanks. And then final question for you at this point, Marina. It's probably more of a general biomarker question. How do the changes in volume on SWI based on iron compare with changes in volume on T1?
The sensitivity is not the same. We know volume performs really well, you know, but we also have a lot of evidence of volume, which we don't have on QSM. The other thing with QSM is that the methods are still developing, and we are now, you know, the field it's a much more recent method compared to volume. So things are still changing with the reconstruction to improve the sensitivity of the metric. But the key thing is that, for example, compared to coded and putamen, obviously volume outperforms QSM. But when you look at globus pallidus, you don't have an alternative. Volume is not an option there. So you have to look at QSM. So it really depends on the endpoints. It's not so much a question of it's not just about power. It's also about what the region is.
Right. Great. Thanks. There's actually, Jeff, I'll come back to a more general question for yourself. I think you briefly mentioned on your slides external control groups. And there's a question around that. What role do you expect external control groups will play across the various trial designs you have outlined? And I guess what I'm adding is that this might be specifically relevant to more complex modes of administration that we're now seeing that may require surgical intervention, for example.
Yes, I think in the near future they're going to play a large role. Even if you have randomized controls, you can supplement that control group with external controls, which can sometimes dramatically increase your power to detect treatment group differences. So certainly, even with the small molecule therapies, I think that it's going to be important, but especially with advanced therapies where you're going to have a small number of treated individuals, and you probably are going to want to match to a larger external control group. I would emphasize that if possible, to have a concurrent control group would be the ideal. CHDI seems to be open to this concept, and so I would encourage sponsors who are planning trials to get in touch with them, because it would be great to have a natural history cohort that is measured with the frequency of visits that are planned for the clinical trial, for example, and certainly in the same calendar time.
Okay, thanks. There's there's a question that is quite specific and to one particular development program that I think might be linked. So I'll put it out here, and I'm curious to hear your thoughts. It's on the AMT 130 trial that I think you mentioned as well on your slides, Jeff. Reading out the question, I was curious both of your thoughts on AMT 130. Obviously, the US regulator so far keeps going back to the one-year data on the randomized controlled trial where they had issues. From your experience, what is the chance of these 3-year results being truly placebo? Seems the TFC, from my understanding, would not have placebo at 3 years, and that number was 60% slowing. Does that make sense?
Yes, I agree. I mean, after 3 years, I wouldn't expect there to be a placebo effect. But I do have to caution people that the graph that I showed with those means does not model a placebo effect for the external controls because they come from enroll. So the time saved that I was talking about there, 2 years, may be overly optimistic in that placebo is not accounted for in that. So I think that there's some issues, in interpretation of the trial results as there always are. That's separate from this question of the sham results. And the sham results did show some advantages of being in the sham group at 12 months. And I'm not sure what to make of that, to tell you the truth. It sounds like the FDA is not sure what to make of that at this point. I think we need a larger sample size and maybe a longer sham period to get some definitive information about that.
Okay, thanks Jeff for sharing your thoughts. Anything you want to add on that from your side, Marina? Great. I think we're almost coming up to the end. So I'll just have one quick last question for Jeff, which just came in, which maybe has a simple answer to it or maybe not. You presented time saved with treatment and responder analysis as 2 promising frameworks. If a sponsor came to you today designing an ISS stage 2 trial, which would you recommend as the lead approach and why?
The time saved, because it works at the group level, so you're going to have fewer variability problems. The responder analysis, I didn't have time to get into it. If you're going to score at the individual level, there can be a lot of measurement error, as you saw with the SDMT. And so scoring at the individual level may be more difficult than in the long run than dealing with the group level or the mean level, which time saved at which time saved is the level of analysis.
Great, thanks. Thanks, Jeff. Thanks, Marina. Great discussion, great presentation. We're spot on time, I think. So leaves me handing it back to James. Thank you.
Yeah, thank you. Well, I agree. Yeah, great presentation, discursive and fascinating. So thank you. As Robin mentioned at the beginning, this is the first in a series of webinars we'll be doing this year. There will be another one later in the year in autumn, another one on HD, but there is one in AD and PD coming. The AD one will be in May, the PD one will be in June. So if you are interested, if you found it interesting, please do join us again for the next in those series. And as I said, there's another further HD one coming in the autumn. But thank you for everyone's attention. Thank you for your attendance. The recording will be available after this webinar, so you will be able to go back and review the slides and review some of the Q&A as well. But thank you all for your attendance. Wishing you a good afternoon.
Thank you.